So my project was about the effect of soil moisture on mealworm health. I did this because mealworms are great for decomposition of organic materials and styrofoam and some plastic materials. The way I measure this was I put them in mason jars with 50 milliliters of water, 25 milliliters of water, and 50 milliliters of water. The 15 milliliter water came out the best, which shows if you're going to put mealworms into a landfill, the environment the landfill is in, you probably want it to be a lower moisture environment. And then if you're going to do a compost pile at your house, you do not want extra water in that pile and to keep it as low water as possible.
We tested the effect of different music genres on systolic and diastolic blood pressures, and we did this because of our own individual interest in music and because of the alarming statistics considering hypertension in the United States, stating that almost half of U.S. adults have the condition.
In our project, we had 20 participants in 5 groups: pop, rap, heavy metal, classical, and jazz, and they each listened to their assigned music genre for 15 minutes. We hypothesized that jazz would be the music genre to lower systolic and diastolic blood pressure the most. Our hypothesis was proved correct.
We created 2 graphs, one with the average systolic and one with the average diastolic blood pressure, and we found that jazz lowers both systolic and diastolic blood pressure by 3 millimeters of mercury, which proved our hypothesis correct and states in the end that if a hypertensive person were to listen to jazz music, then it's more likely to help their condition beyond the use of medication than the other 4 genres we tested.
My project was a meta-analysis on time and therapeutic effect between genotype-guided and clinically guided dosing of Warfarin. My hypothesis was that the genotype-guided grouping would have higher time therapeutic effect, and it was suggested that that was correct.
My project is about the effect of various purification techniques on removing dye from water, and we took three techniques using ZIF8, decolorizing carbon and sugar carbon, as well as 3 dyes, Sudan orange, Congo red, and tartrazine, and we saw what dyes were removed best with what treatments. And we concluded that decolorizing carbon and ZIF8 are the best at removing Congo red, Sudan orange, and tartrazine respectively.
I tested the effects of roadside contaminants on wheatgrass germination. I tested this to discover that if, as a society, we should be concerned about the plants along our roads and waterways because they help prevent erosion. I tested car soap, insecticide, and road salt, and I hypothesized that these contaminants would prevent the seeds from germinating due to disrupting water intake, cell structure, and the environment surrounding the seed. The results proved my hypothesis partially correct, as while road salt did prevent any of the seeds from germinating, car soap and insecticide both had a germination rate of 100%.
So in my project, I did early detection of Parkinson's disease using machine learning. I tested 2 different types of machine learning models, a decision tree and a logistic regression model. My goal was for both of them to have F1 scores of 90% or higher, and I found that decision tree had an F1 score of 92% and logistic regression had an F1 score of 95%. I did this project because voice dysfunction is the earliest feature of the Parkinson's disease, and basically we can use these models to help lead to an earlier diagnosis, which can lead to faster treatment and preserving of the neurons in the brain.
So our goal for our project is to build a handwriting robotic arm to teach children how to handwrite because there are a lot more students for one teacher.
And overall, we were successful because we coded 52 letters, uppercase and lowercase, to perform the motions of handwriting, which children can follow along and learn.
So our project was called Removing Pollutants from Water using modified sodium alginate beads. And our additives to the beads were activated carbon, bentonite clay, cellulose nanofibrils, and iron oxide nanoparticles.
The activated carbon did the best at removing both methylene blue and Congo red from the solutions. After 24 hours in the solutions, they were clear and had no dye left.
Okay, so we tested the effect of different cocoflavanol concentrations on P. fluorescens biofilm formation and quorum sensing, and we did this because P. fluorescens is a common bacteria found in milk and milk processing equipment.
We found in the 1% pretreatment that there was less biofilm formation, bacteria growth, and it reduced the expression of one quorum sensing gene.
My project was determining which cryptochrome gene in A. thaliana exhibits the most growth when in the presence of a magnetic field. So for my project, I had wild-type Arabidopsis plants, cryptochrome 1 gene alteration plants, and cryptochrome 2 gene alteration plants. Overall, in my conclusion, I found that cryptochrome 1 gene plants had the highest growth rate, while the wild-type, aka control, had the best health. In the future, I hope to do genetic combinations with this project and see where that leads me.
My science fair project was on the effect of prompt style on answer usefulness. In my project, I tested 3 different types of prompts. Prompt A, a standard prompt, prompt B, a creative prompt, and prompt C, an informational prompt. I hypothesized that prompt B, the creative prompt, would be the highest ranked and rated by the reviewers. My hypothesis was correct because prompt B was the highest ranked and rated by both reviewers.
My project is the dissolution of bath bombs with different amounts of cornstarch. Generally, people would like bath bombs to dissolve slower because it increases the scent, bubbles, and color. My hypothesis is that if more cornstarch is added to a bath bomb recipe, then it'll dissolve slower. I made one bath bomb with 6.5 tablespoons of cornstarch and another with 4 tablespoons of cornstarch. I placed them both in water at the same time and then timed their dissolution rate. In the end, there was a significant difference between the 2 times. The average of the extra cornstarch bath bomb, which was 6.5 tablespoons, was 44 seconds, while the normal bath bomb, which was only 4 tablespoons, was 12.66 seconds.
My project is called The Effect of Homemade Filters on How Clean Water Will Become, and I chose this project because I wanted to learn how, if you were stranded or hiking in the woods, if you would be able to filter water using fabrics. And I chose these fabrics because you would most likely have them while you were in the woods. My conclusion stated that the canvas fabric filtered the best. And I also learned why hiking socks are wool and not cotton, because it doesn't absorb the water.
My project is about testing different circuits at different temperatures. My project proved that the conductors at the superior is copper. Also, it proved that wires work the best at the lowest temperature possible. The battery itself was working the best at the room temperature, and the circuit itself was working the best at the lowest temperature possible.
My project is what is the effect of different mass distributions on the total spin time of a 3D printed top. My hypothesis in this project was if center mass distribution is used, then the top sample will spin for the longest amount of time on average. In this experiment, I evaluated 6 different tops, 5 with different mass distributions, and 1, a control group with no mass distribution. I gathered several conclusions from this experiment. First, bottom center mass distribution top sample spun for the longest amount of time, and one side mass distribution top sample spun for the shortest amount of time.
My project is the effect of artificial intelligence algorithms on sentiment value/score accuracy. My hypothesis is if ChatGPT is used, then the sentiment value will be most accurate. The data shows that ChatGPT did have the highest percentage after a human, meaning that it can understand and use human sentiments.